Okay, so welcome back to Chapter 7. We're going to be talking about membrane transport. So there's different types of ways that things can be transported across the membrane, and some of it requires proteins. If you remember from the first segment, I spoke of this figure right here showing the membrane, the hydrophobic tails, and the protein that spans both sides of this membrane, and you can see all the water molecules. There are two major types of transport across the membrane. There is active transport and passive transport. Active transport requires energy. Passive transport does not require energy. We're going to start with passive transport. Passive transport uses a concentration gradient to move solutes along their concentration gradients. So basically the concentration gradient along the concentration gradient is going down the concentration gradient. So it's moving from an area of high solute concentration to an area of low solute concentration. Passive transport does not require energy at all, and it can occur with or without the assistance of a protein. Now, it's important to talk about diffusion in general because we're going to be expanding upon that. So simple diffusion is basically the movement of solutes from an area of a high concentration of solute to an area of low concentration. So it's like going downhill. So there's other ways to say this. Okay, so you could say that these solutes are moving along their concentration gradient, or the solutes are moving down their concentration gradient. So there's a number of ways to say it. It all means the same thing, meaning that the solutes move from an area where there's a high concentration of solute to an area where there's a low concentration of solute until they reach equilibrium. So we're going to talk about equilibrium. Let's look at these two examples right here. So if we look at this figure at the very top, we've got orange dye molecules on one side of a membrane. And then we've got this membrane which actually allows, the pores are large enough to allow the molecules of dye to move across it. And then if we look at the bottom, we've got orange dye molecules on the left and purple dye molecules on the right. Now, it's important to state that diffusion can move in both directions at the same time. So the molecules are going to be randomly moving, and they're going to move from, look, we've got on the left a high concentration of the solute, okay, of the orange. And then basically the orange are going to move at a rate which increases from left to right until they reach equilibrium. And the same thing goes for the bottom, right? So the orange are going to move to the right and the purple are going to move to the left, right? So it's just the rate of diffusion is increasing from, in this case, the orange from left to right and the purple from right to left. So if we look at what's happening over time, they are moving back and forth. It's not just one direction. So they're going to move back and forth, but the net movement is towards the lower concentration of that particular solute. So let's say they've reached equilibrium now, or dynamic equilibrium. At that point, the rate of movement across the membrane for both of the solute molecules at the bottom and just the orange at the top is the same, going to the left and to the right. So they can still move across the membrane. They're just not static, but they are moving from left to right at an equal rate when they reach equilibrium. And this is called dynamic equilibrium. Now, in terms of water, water diffuses as well. When water diffuses, it's called osmosis. Now, water diffuses from an area of low solute concentration to an area of high solute concentration. So here we're talking about the rate of movement of water, not the solute. So solutes move from high to low. Water is going to move towards the higher solute concentration. So basically it moves from low to high in terms of solute. Water a little bit more. So if we look at this U-shaped container right here, and there's a membrane, right, separating the two sides. So on the right side, there are more molecules of these green molecules than the left. and there's an equal amount of water. Okay, so based on the definition that I just told you, which is water is going to move from an area of low solute concentration to an area of high solute concentration, the water should move to the right, towards the higher solute concentration right here. So if we look at what's happening at the membrane, we can see that the water is allowed to pass the membrane, but these green solutes are not. Right? So if the green solute molecules were able to cross the membrane, then it would be equal, right? So the green would move to the left and water would kind of equilibrate as it's moving. However, only the water is allowed to pass through the membrane. So the water is going to move towards the right at a higher rate than it is going to move towards the left. So if we look at what happens over time, we can see what's going to happen, right? So I already described that the water should move at a higher rate towards the right, and the green solute molecule is not allowed to cross the membrane, so that's not going to move at all. So at equilibrium, what we want is the same amount of solute per volume of solution. So if the water moves to the right until it reaches equilibrium, the right side of this U-shaped container is going to have a lot more solution in it than the left side because there was more solute there. Right? So since there was more solute molecules, more water needed to be moved over to that side in order to make the concentration, which is the amount of solute per volume of. Now, we're going to talk about tonicity, and this is kind of related to typically a cell and its environment. So tonicity is the ability of the environment solution, right? So the surrounding solution around a cell to cause a cell to either gain water or lose water. It depends on the concentration of solutes in the cell and in the environment as to which direction the water is going to move. Is it going to fill the cell, or is it going to come out of the cell? Or is there going to be an equal rate of movement in and out of the cell? So there's terms for this. We have an isotonic solution, is a solution in which a cell is in which the solute concentration is equal to the solute concentration inside the cell. A hypertonic solution is a solution that has a greater solute concentration than inside the cell. And a hypotonic solution is a solution that has a concentration of solute that's less than that of the cell. Let's look at what happens to different types of cells in each of these conditions. If we look at this right here, we've got an animal cell at the top and a plant cell at the bottom. Now let's talk about some characteristics of animal cells and plant cells and the differences between them. So these are both those cells as showing as they would normally be shaped in an isotonic solution. So in an isotonic solution, the rate of water movement into the cell and out of the cell is equal. So the cell's kind of happy. It's not going to take on too much water, and it's not going to lose too much water. Okay, so this is a blood cell. This is representative of an animal blood cell. And animal blood cells are normally like donut shaped. Okay, so they're smooth and donut shaped. And then, of course, plant cells are generally somewhat flaccid or just regularly shaped. They're pretty happy. They're not filled with too much water. Now let's look at each of these cells in a hypotonic solution. So remember, a hypotonic solution is a solution that has less solute in it than the cell. So if the hypotonic solution has less solute in it, that means it has more water. So the water is going to move into the cell towards the higher solute concentrations. Remember, inside the cell, the solute concentration is higher than in the hypotonic environment. So the difference between plant and animal cells, remember, plant cells have a cell wall. Animal cells do not have a cell wall. So if an animal cell takes up too much water, it will become lysed. Okay, so it'll break apart is what that really means. So it'll burst. A plant cell can take on a lot of water, however, it's regulated and it's controlled by the presence of that cell wall. So the cell wall keeps the plant cell from taking on too much water. So the plant cell becomes turgid. And in a hypertonic solution, this is a solution which has a higher solute concentration outside of the cell than it does inside of the cell. So if there's more solute outside of the cell in an animal cell, or at least a blood cell, it's going to become shriveled up. Okay, so it's going to lose water because water is going to move towards the higher solute concentration. In a plant cell, the cell wall maintains its shape. However, the plasma membrane peels away from the cell wall, and basically, because it's losing water, right? So all the water is leaving the cell, and it peels away, it shrivels up inside there. Okay, so this is called plasmolysis. So you could say that this plant cell has been plasmolysed. Let's look at some other things. So we've got this organism right here. It is a paramecium, which is a protozoan. These types of cells live in fresh water, which has a very low solute concentration relative to the cell. So their environment of organisms can regulate the amount of water that goes into the cell. And they can regulate it by this contractile vacuole, which you should remember from chapter 6. Okay, so this contractile vacuole is going to take on a lot of water, and once it takes on enough water, it's going to contract and then basically expel the water out of the cell. Now let's look at this video of it. So take a look at it. You can see the cilia, which allows it to move around. So I'm going to kind of point it out as it takes a couple minutes, and it's very, very hard to see. So you can see that right here. Whoops. Let's play it again. So right here, oh, you see that? It's very, very faint. It's going to happen again. So opened up, it's filling up with the water, and then it's going to... Let's look at inside plant cells. So this is a plant cell in a hypertonic solution, which is a solution that has a higher solute concentration. So the water is going to leave the cell. So let's see. We can see the chloroplasts moving around. Okay, so the water is leaving the cell, and you can see the plasmolysis is occurring. So you can see the plasma membrane is peeling away from the cell wall, and all the cell contents are getting squished up because all of the water is leaving the cell. Now let's look to the right. In a hypotonic solution, this is a solution which has less solute than inside the cell. The cell's going to take on more water. Okay, so again, you can see the chloroplasts moving around. This is called cytoplasmic streaming. They're going to take on more water and become more turgid. So the cells are going to be very stiff, right? All right. So here, what's happening is the excess water is going to be placed into the that central vacuole, which fills up. So that's just plain diffusion, and of course diffusion of water, which is osmosis. Let's talk about facilitated diffusion. Facilitated diffusion is passive transport. Again, passive transport does not require energy, but with the help of proteins. Okay, so transport proteins are required in facilitated diffusion. There are two types of transport proteins involved. And carrier proteins. So let's look at the difference between them. So channel proteins, some examples are aquaporins, ion channels, and gated channels. Channel proteins just provide a channel, right? So whatever molecules can fit or whatever is allowed to pass based on its charge and, of course, size will be allowed to pass through this channel. And they're going to move from an area of high concentration to an area of low concentration until the rates or until both sides are equal in concentration of that solute, typically. Okay, so channel proteins just make a channel. It's like a pathway. Carrier proteins are a little bit different. They almost like pick up. They're carrying the solute, right? So they pick it up. You can imagine maybe it picks it up with its hands, but it really doesn't have hands. It has what's called a binding site. Okay, so we've got a binding site for this molecule right here. So carrier proteins will be present in an open conformation and a closed conformation, depending on what side of the membrane that we're dealing with. So here on this side, on the extracellular side, it's in the open conformation. So if there's enough of this solute molecule that actually binds to this carrier protein, it's going to enter there and then bind. Once it binds, the carrier protein changes shape. And it's going to change shape and basically close on that side and then open on the other side so that the solute particle or molecule will move from the extracellular side to the intracellular side of the cell. And that could go the other way. It depends on which side has the higher solute concentration. So in this case, the extracellular side has higher solute. It's going to move from an area of high concentration to an area of low concentration. Okay, so if you remember the first slide, I introduced you to the two types of transport. We just talked about passive transport, and remember, there's also active transport. Active transport uses energy to move solutes against their concentration gradients, not along their concentration gradients. Transport proteins are required for this. So in all cases, there's some sort of carrier protein that is involved in this process. Active transport actually allows the cell to maintain a concentration gradient that may differ from its surroundings. Now, it could be less or more, right? So they're not going to become at equilibrium at any point in time. Okay, so this maintains solute concentrations different on both sides of the membrane. Again, active transport, the proteins require energy, and this is usually coming from the energy from ATP hydrolysis. And again, carrier proteins are always involved. Let's look at an example of a active transport process using the example of a sodium-potassium pump. So as we can see, we have the membrane here. This is the protein, and on the inside of the cell, we have a low solute concentration, in this case sodium. The sodium-potassium pump actually transports 3 sodium out of the cell and 2 potassium into the cell. So you can see inside the cell there's a low sodium concentration. However, we're still moving sodium out of the cell. So again, this is against the concentration gradient. So sodium is moving from an area of low concentration to an area of high concentration. So the first step is the binding of sodium. The next step is the hydrolysis of ATP. So ATP is hydrolyzed into ADP and inorganic phosphate. The phosphate is going to be attached to the protein. So in this case, the protein is said to be phosphorylated. When a protein becomes phosphorylated, it's going to act different and it may change shape. Okay, so what's going to happen is it's going to close on that side and then open and allow the sodium to go towards the higher sodium concentration. So it's going to go against its concentration gradient. While it's open on the outside, this is when potassium can bind. And remember, if we look at the first box here, potassium is low on the outside of the cell, and potassium is going to be moving towards the higher solute concentration. So again, potassium is also moving against its concentration gradient. So potassium binds, and then the protein becomes unphosphorylated or dephosphorylated. So what's going to happen is this phosphate is going to be removed from the protein, and then ATP will be made again because that phosphate, inorganic phosphate, will combine with ADP in order to make ATP in the ATP cycle. But here we can see that potassium has bound, and then the phosphate has been removed so that the protein moves back to its original shape. And then it can complete that same cycle again. Now it's ready to bind more sodium, and keep doing this until it doesn't need to anymore. So let's look at
Sometimes a
this
cell
process
needs to move a solute against
just
its
in an
concentration
animation
gradient.
type format.
This process is called active transport, and it requires input of energy from ATP. For instance, most animal cells need to expel sodium ions, Na+, and take in potassium ions, K+, both against their concentration gradients. Here is how the sodium-potassium pump works. Sodium ions bind to a transport protein. ATP transfers a phosphate group to the protein, providing the energy that causes the protein to change shape and push the sodium ions across the membrane, where they are released outside the cell. Potassium ions now bind to the transport protein, and the phosphate group is released. This causes the protein to return to its original shape, releasing the potassium ions inside the cell. The transport protein is now ready to repeat the process.
Okay, so that's pretty much what I discussed, and now let's kind of review what I just went over. So remember, we have simple diffusion, which is just movement of solutes across the membrane, and this can happen with very small and hydrophobic solutes. Okay, so they don't need a protein to help them. And then we have facilitated diffusion, and this can be done by channel proteins and carrier proteins. And again, remember, this is still passive transport. The solutes are moving from an area of high solute concentration to an area of low solute concentration. And then we have the active transporter. The example given in your textbook is the sodium-potassium pump. However, there are others that also do the same. And remember, active transporters require the hydrolysis. All right, so let's talk about the membrane potential. All right, so we're going to talk about ion pumps and move into the proton pump. Membrane potential is the voltage across the membrane, and this is generated in by a couple different forces. And the forces collectively together are called the electrochemical gradient. The electrochemical gradient drives the diffusion of ions across the membrane. Now, what is the electrochemical gradient? Let's talk about that. So we've got a chemical force, that's the chemical gradient part, and then we have the electrical force, which is the electro part of the chemical gradient. Now we have the chemical force is basically a concentration gradient. And what that means is that the concentration of the solute is higher on one side of the membrane than it is on the other side of the membrane. So that creates a gradient. And then we have the electrical force. This is basically the effect of the movement of the ions. In some cases, the ions, of course, may be positively charged or negatively charged, and this creates an electrical difference or like a voltage across the membrane. Okay, so basically you're going to have a more positive side of the membrane versus a more negatively charged side of the membrane. Now, an electrogenic pump is what generates this voltage. An example of an electrogenic pump is a proton pump. Electrogenic pumps help store energy that can be used for the cell to do work. Now let's look at the proton pump. So we have the proton pump. It's attached to the membrane, and there is a space where the protons can be pumped from, in this case, from inside of the cell to the extracellular fluid. Again, ATP is required. So here we've got the chemical gradient being formed. You have a higher concentration moving against the concentration gradient. Remember, this is an active transporter. So we've got a higher concentration outside of the cell of protons, which makes that side more positively charged, and then the inside of the membrane more negatively charged, which is that electro part of the electrochemical gradient. Co-transport can depend on proton pumps and these types of electrochemical gradients that are being generated. This is how the electrogenic pump can be used to do cellular work. So here we've got the proton pump again. We've got protons being pumped against a concentration gradient. This is active transport. This concentration gradient can be used to drive passive transport, right? So we have active and passive working together. This is called co-transport. So here we've got, it's actually a symporter because it transports two things in the same direction, right? So we have it's same sim. Okay, so sometimes there are antiporters, so they could transfer two things, one in one direction and one in the other. However, this is a symporter and this is a passive transporter, which is actually using the chemical gradient that is generated from the active transporter. So here, these protons are going to move back from the outside of the cell back to the inside of the cell based on concentration gradients. So here we've got higher concentration to lower concentration, passively transported. At the same time, this allows the transport of sucrose into the cell. Okay, so that's an example of co-transport. And co-transport is basically active and passive transport working together. Now, this kind of summarizes some interesting figures for the membrane proteins. So this shows actual space-filling diagrams of the membrane, right? And then we can see what the proton pump looks like. We have a calcium channel and an aquaporin. Remember, those are specific for moving water into and out of the cell. Okay, so these are all proteins right here. These are all membrane proteins that function in transport. Now remember, transport isn't the only function for membrane proteins. Some of them can serve as signaling molecules as a receptor, right? And some of them can be enzymatic. So some of them can help catalyze chemical reactions. All right, so let's talk about bulk transport. Bulk transport is a way to move lots of small molecules and water into the cell, or maybe out of the cell, right? And also large molecules like polysaccharides and proteins. So there's two major types of bulk transport. There is exocytosis and endocytosis. Both of them require energy. So these are forms of active transport. Exocytosis, you can kind of think of it like exiting the cells. So things are being exported out of the cell. Endocytosis is into the cell. Now let's exocytosis. Well, all of them kind of involve vesicles, but exocytosis involves transport vesicles that are going to migrate to the membrane and fuse with it and then release the contents from the cell. There are certain cells that actually do this regularly. They are secretory cells. Endocytosis is basically just the opposite. So the cell takes in macromolecules and other things by forming vesicles at the plasma membrane. So endocytosis is just basically the reverse of exocytosis, but involves different proteins. There are three types of endocytosis. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. Phagocytosis can be nicknamed like cellular eating, and you'll see why in just a moment. Pinocytosis is like cellular drinking, and receptor-mediated endocytosis is just something altogether special. So let's look at phagocytosis. Phagocytosis is when a cell engulfs a very large food particle. So it's, remember, cellular eating. What actually happens after that is it's like a vesicle, but it's larger, so it's called a vacuole. Okay, so a food vacuole forms. This food vacuole will eventually fuse with a lysosome, and then remember the contents of the lysosome, right? So that contains digestive enzymes and a very acidic pH, so it helps break things down. So there, we're eating and we're breaking the food down. Pinocytosis allows the cell to create vesicles along the surface of the membrane to take in small molecules and water. So it's like drinking, right? So what forms with pinocytosis is Uh, they're like coated vesicles. They've got proteins that coat the vesicles, and they're handled in a different way. Receptor-mediated endocytosis is very similar to penocytosis, except a receptor is required. So here we've got this little red receptor that looks like it's shaped like a Y. So it has a special shape. So the only thing that can bind to it is whatever fits in that little V part right there. So these little purple triangles fit in that. So those can be called what's called the ligand, Ligand. Ligands are just molecules that bind to receptors. So once this binds to the receptor, it induces the formation of a coated vesicle. They're usually coated with a protein called clathrin. Clathrin-coated vesicles, again, are handled differently than regular vesicles. So again, this allows the take-in of specific molecules. So if we think of phagocytosis and penocytosis versus receptor-mediated endocytosis, the difference is that receptor-mediated endocytosis is very specific. The others are more general. They just take in whatever, right? This is, it's taking in a specific molecule that binds to that specific receptor. All right, so this kind of summarizes bulk transport. So we have phagocytosis, cellular eating. A food vacuole forms, which is going to eventually fuse with a lysosome to break down that food. Here we have coated pits formed with penocytosis, except this is more like cellular drinking. So water and smaller ions and molecules are going to enter the vesicles and be taken in by the cell. And then receptor-mediated endocytosis requires a specific interaction between a receptor and a ligand or a molecule that binds to it. Once it binds, it forms these coated pits, which will be handled in a different way in terms of metabolism. This summarizes transport. So remember, there's active and passive transport. Passive transport involves just simple diffusion. Part of that would be osmosis and facilitated diffusion, and I talked about tonicity. And then active transport, we've got the sodium-potassium pump, the proton pump, and then co-transport, and of course bulk transport, which involves exocytosis or exiting the cell, or endocytosis or taking in things by eating, drinking, or something specific. All right, thank you for listening.